So today we're going to be going through two senses. It took quite some time to go through sound in detail and then go through sight in detail. Today we're going to be going through two senses, which are smell and taste. And they are actually, I would say, much harder than sight and sound. We can go through them in one day for two reasons. One, we've already gone through two of the senses in quite some detail, and as you're going to see today, even though they work very differently, the senses of smell and taste, sense things on one end and send electrical signals on the other end to the brain. So there are some things that are in common. The other reason is that we as humans and scientists really we just don't know enough as much about the senses of taste and smell as we do about sight and sound. And we'll get into some of the reasons for that today. The quote that I have for today is, "A rose by any other name would smell as sweet," and that's by William Shakespeare. And I think those dates are right, but at least I have him passing away sometime after he was born this time. There is going to be required reading. We're going to move along in the chapter. Hopefully you've had a chance to at least start chapter 2, if not finish it. This is going to be the last required reading that can be on the first exam. So if once you've done this reading, if you've done all the other required readings, including that one required reading that was a video, then you'll have done all of the required reading, but that's going to be on the first exam. There's one more class day we have before the next exam, and so that can be on the next exam, but there won't be any more new required reading. Okay, so I wanted to remind everybody of upcoming important dates. We've actually stuck to the prontuario in terms of the due dates for the homeworks. And we're going to do that moving forward and also for the exam. Homework number 3 will be available on Moodle before midnight tomorrow. There is a chance it may be available on Moodle before midnight today because at least the university was closing. I got to spend some time early this afternoon finishing the homework number 3 up. So it looks like I'll actually be able to get that online today, but I wanted to have a date for certain that I could commit to, and that date's by tomorrow at midnight. So just like last time, after I put it on, I'll put a note in Piazza. So I know this is a long weekend unless for some reason the university changes that. So I'd like to get it up today if possible to give you a head start to get it done. Okay, the due date has not changed. It's going to be on Wednesday, February 20th, which will be 1 week from today. This Monday, February 18th, is a holiday, as I mentioned, so I'm not going to have office hours on that day. But if you do have questions, I'm pretty much available almost every day. So put your questions on Piazza. The sooner you put them, then the more assured you can be that I'll be able to respond to them. Exam number 1 is going to be given on Monday, February 25th, which is the same date that was shown in the prontuario. That is one exception. If there is some reason why we can't get into the classroom on that exam date, then that class would be canceled. So you wouldn't come on Moodle because there wouldn't be no class just for that date. If it happens to be canceled on that date, because at least part of the exam is going to be in the classroom and you're going to be writing things on paper. So because you're going to be writing things on paper, please bring pencils or pencils and pencil sharpeners, or the part that will be given out in class will actually be given out on a printed piece of paper. There is likely going to be an online portion that you would be able to do at home. That portion, if there is going to be one, will be available online before midnight on Thursday, February 21st. So that's a week and a day from today. The online portion of the exam would be available online to the extent that there is an online portion. My intent is to do both, but if I have not given you the online portion by that time, then everything will be in the classroom on February 25th. My hope is by giving you some of it online before, we can make the portion that will be given in class less time consuming, or give you more time to do it. The material that will be on exam number 1 is going to relate to everything that we've covered in the classes. So far, all of the classes I've been successful in recording. So the recordings from all the classes are online. Although I do realize that sometimes it's hard to understand what I'm saying on sometimes during some of the recordings. All of the classes are also online. So far we've been successful in recording all the classes. Remember that that's not a guarantee. If something messes up, and then that class wouldn't be available online, but the slides still would be. And then all of the required reading, including that video that's been assigned prior to the date of the exam. And that's basically everything that's been done already. So there could be material on the exam that's covered in the next class, but there wouldn't be any material on the exam that would be more reading because I've already just now assigned the reading. So if you're getting through chapter 3 again, then you'll have done all of the reading. I suggest that you review the class slides carefully, and for parts of the slides that you're unclear on, then just go to that part of the class video, and definitely do it sufficiently before the exam so that if you're confused about something, you can ask me in Piazza or office hours. If next week before the exam or the week after, if sometime before the exam you would like to meet with me or want me to schedule a review for you and others, just feel free to ask that in Piazza. But my hope is that if you go through all the homeworks, if you go through all the class slides, if you paid attention in class, that the exams are going to be pretty straightforward, although I do intend them to be comprehensive. So there's going to be things in the exam related to modeling, related to emotions, related to influence tactics, related to senses. So the exams will be fairly comprehensive. Any questions on either homework number 3, exam number 1, or any other logistical kind of issue before I jump into today's topics? Okay. And as usual, if you do come up with something, either put it in the chat, or if it's something you want me to stop, go ahead and just speak. Quickly in review, want to remind everybody that the environment is reality. There are 5 types of sensible energies or interactions. Forces and electromagnetic energy were what we've seen examples of that with our hearing that's sensitive to vibrational forces in the air. And then our sight, which is sensitive to electromagnetic energy. Today, as we go into smelling and tasting, we're going to be talking about biochemical interactions. The human's eyes and ears only detect a very small part of what's photonically or sonically sensible of reality, and electronics can do much better at sensing outside of those ranges. Okay, so there's going to be an interactive part. There's two of these interactive slides today. And I assume everybody can see the chat. I'm actually not sure that's the case. I'm going to put it here. It might be too small. So yeah, it's like I can't, it's not actually something you can see on the screen. Well, so what we're going to do now is I want to make sure that you understand sort of what's the commonalities and the differences between sight and hearing. And this is going to be more important at the end of today. If we have time, we're going to go through the differences between sight and hearing and taste and smell. So on the left here, upper left, I have ears and sound. So the ears work a certain way, and they allow us to detect the phenomenon of sound. And then the eyes work a certain way, and they allow us to detect the phenomenon of light. So what I'd like you to, we're going to start here is say, okay, it could be either how our ears and eyes similar or how our sound and light similar. I want to put some of those similarities here, and I'd like your help to do that. So just feel free to put some similarities into the chat, and I'll put them here, then consolidate them here together.
Yeah, I would say one thing that they are alike is that they have some kind of physical organ that will receive. Yeah, like Maria said, that they both take in input, and it's through a physical, organ that it is later translated to, electrical signals that our brain processes.
I'm going to add here in the ranges, I'm going to put frequency ranges in parentheses. But I guess there's also intensity ranges. So maybe I'll put that in a separate line. If you're not feeling inspired for a fifth one, we can go back to that one and start working on some of the differences. So here, and you can go to the left or the right, just tell me which one you're working on. So this would be a characteristic, for example, of ears or sound. that we would write here on the left that would then be different than a characteristic of eyes or light that we would write down on the right.
Let's say ears and sound work with vibrations. They interpret vibration, and then whereas light is not, there doesn't have to be physical contact for your brain to analyze it. Yeah, exactly. I thought about it later.
I want it to fit here, but I don't make the font small. Okay. What's a characteristic of the ear or of sound? It's not a characteristic of eyes or light.
Actually, Professor, to add to the things that they are alike, I would add that how we perceive it, how humans perceive it, to perceive sound, you need multiple parts in your, ear, and then eyes, you need multiple parts too. It's not like one organism, one part of your body does everything. It's more like a group of you know, consequent actions that take place in your body that make you interpret both.
I'm not sure that's the best way to write it. I write it like that because we know that for sound, for example, as you go down the cochlea, the frequencies that you're sensitive to vary. And so initially, at the beginning of the fluid interacting with the cochlea, it's one frequency of sound. And as it winds its way down, those parts of the cochlea are different to another frequency of sound. And so there's distinct parts, really, of the cochlea that are sensitive to different frequencies. Yeah. But on sensitivities, there's not really that way. The eardrum is picking up all of the vibrational intensities at all different frequencies.
Yeah, an analogy I just thought of is, think of Legos and each individual piece, how they make something bigger. If you are missing, let's say we analyze ears to be, something you build with pieces of Legos, and you're missing one part of that whole thing you're doing, then you might not hear certain frequencies. So what I'm saying is, like, everything works. They're complex organisms. It's not straightforward. Not one piece of Lego does everything.
Yep. Okay. And then just finishing that thought on the eyeball, the cones are really, can be sort of mixed, right? In cases where you have a lot of light, you're going to have red cones. I'm sorry. Red cones, green cones, blue cones. And then you're going to have rods in all sorts of different places. Although it is true that you don't have a lot of red and green and yellow cones the further away you get from the center point. So it's what you're, on the flip side, what you're distinguishing there is different trajectories of the light that's coming into your into your eye. So in hearing, you don't have that. So that's a difference. Let me put some of your differences here now.
I have.
I see one that says, light works a lot together with reflection while sound goes through objects.
I was literally about to say that.
Someone wrote it here. I'll call it traverse objects. It's not really that it's going through the object, unless it's something thin like a drum or something like that. But I see what you're saying. It could also be like a wall that's made to vibrate. You're not going to get light through. So light can traverse objects. And then on the other side, I guess, maybe the counter to that would be light can traverse transparent objects. Let me know if you have any objection to what I write. So there's a comment here that light, sound are waves, but light is more complex because of the particle wave terms. So I have to be careful here. In air, we don't think of sound as particles. So I'm going to characterize that in air, sounds are waves. And then on the other side, I'm going to put light is waves and particles. And the reason for that is because it actually turns out that sounds are also particles when they're in and this is beyond, you don't need to know this for the course, but sounds are also particles when they're in regular solids. And so if you have a regular like solid structure, the way that sound actually propagates through that can also be characterized as a particle. And that particle, by just so you know, is called a phonon. So instead of a photon, it's called a phonon. But phonons don't exist in air because there's no regular pattern within air. And that's, by the way, why sound can travel faster in solids. Okay, let's see what else we have. Okay, we need 2 more on each. And they don't have to be opposites. I'll put one here. With the eyes, they're used to create images. That's one of the things we talked about a little while ago.
I was thinking if they have any difference of dimensions, like with light, you can sense depth, but I don't think with sound.
Yeah, that's actually kind of interesting that you say that, because without your brain, right, and it's a little bit hard here because what I've done in the course is talk about what we sense first and then talk about what we interpret later. And the reason for that is like in the McGurk effect, where some of our interpretations combine senses and we just don't realize it. I don't want you to think as we go through here that when you see, your brain's just interpreting things that are coming through your eye, because that's not true. And when you hear, that your brain's just interpreting things that are coming through your ears. That was what we saw in the McGurk effect. But there's examples of that in other cases. In today's class, when we go through taste and smell, you'll see that things taste differently when you can't smell. So this idea that the different senses are, that your brain basically decides how it wants to combine the different senses to figure things out, that is an important one. There's a comment about emotions. Actually, there's two comments here. One is, with sound, you can triangulate where it's coming from. You can a little bit. Some people are more talented than other people. Your ears are basically going out both sides of your head. Sometimes if it's a very faint sound, you might cover one ear and then uncover that ear. Or you might cover an ear and start rotating your head. Dogs can actually rotate their ears some without rotating their heads. So animals that rely more on their hearing to figure out where sound is coming from will do that. So I'm not sure how to write that particular one. To think about it a little bit. You can definitely use your ears to figure out, to some extent, or to try to narrow down where something's coming from. So, but I guess you could use your, so maybe the person that wrote the triangulate, just anyhow, if you can explain that a little bit more. I think I'm not following based just on what you wrote. Okay, thank you for explaining that. It's probably a little too wordy, but I think that's good. Some of these are, by the way, will be useful for the homework. So I'm going to put up the slides with these inputs just like they are here. We have one more. Hold on. Can we say something? I'm not sure. I'm not sure about that actually. It's a good question. The question is, once the light gets to your eye faster from wherever it left than the sound does. So I guess if we think of the eyes as where it's absorbed, so the eye goes through to the back. Well, I think what we can say is that what you perceive from photons is closer in time to reality than what you perceive from hearing. So I'm going to put that over here as a number 6 because we have space. Closer to reality time-wise. So, if you think in, if you see someone at a far enough distance talking, you can see them, but you're going to hear them later. So what they actually said at the time they actually said it is going to be different than the time you saw them and the time you heard them. But because light travels so much faster, the photons that are coming from that person to your eye took less time to travel than the sound. That's also going to be a really important observation because we've said that the environment is reality, and to observe reality takes time, and we're in some space, and so light is about the fastest way we can observe what we can about the environment. Okay, let's move on. We took some time there, but that was really helpful, and I say that because some of the things that we just talked about I know are related to the homework because I've been working on it. So, okay, so observations of human senses. Some of these we've talked about already. Hearing and seeing kind of go together. Sounds are vibrational phenomenon of varying frequency and strength. Light is electromagnetic phenomenon of varying frequency and strength. You can think of strength as the energy of light, and energy of light is proportional to the frequency. The resulting electrical impulses make their way to the brain and are pretty well organized in space and or time. Light at the outside of an image that you're looking at is coming from a certain part of the cornea. And if it's in a low lighting environment, then it's coming from rods that are in that part of the cornea. And if it's in the center of the scene, or if it's in a bright environment, then it's coming from rods or cones. In a bright environment, mostly cones that are located around the center. And as light comes in, those interactions are make it pretty quick to the brain where they're processed pretty quickly. Same thing really with sound. Sound is it's coming in, it's vibrating, and there's a sort of train, if you will, of signals that are coming to the brain. So both of them give you high-speed, high-quality data that I would say the brain really doesn't have to do much to in terms of processing an image. What the brain then can focus on in sound and sight is processing what's in the image, right? Or what words are being said. If it had to sort of spend time deciphering the sound to put it in some ordered regime before it can interpret it, it would be much less effective than it is. Okay. Here's why I love eyes and ears. I love eyes and ears because they're photons and phonons, although they're not really phonons because mostly we're talking about in air. It's physics. There are a limited number of variables. So in light, we have the location that it's originated from, the photon that's come hit your eye. We have its frequency and we have its intensity. And for sound, it's even easier. We just have the vibrational frequency and intensity. There's one relationship, and that's given by the physics. And those equations are known. And those physical phenomena are transduced into electrical signals. And we can, in sensors, we can take those signals and put them through an A to D converter and computerize them, turn them into digital data. Frequencies allow us to perceive lots of information about what's transmitted simultaneously, so we can actually separate frequencies out. Smelling and tasting is totally different. Smelling and tasting is biochemistry, not physics. So in tasting, we're going to talk now about how our tongues sense, although there's actually taste receptors in our mouths and in the back of our throats. So it's not just in our tongues, but mostly in our tongues. We'll talk some about resolution and diversity. You'll see how that's different than resolution and diversity in sight and sound. We'll talk about sensitivity and artificial tasting. So what does an electronic sensor look like when it's tasting? And then we'll do the same thing for smelling. If we were in the classroom, I could have brought in some popcorn so you can, and lollipops, but we'll have to do it virtually because we haven't figured out how to taste and smell very well over the internet. And then next time we'll talk about touch and internal sensing. And then I'll also reserve some time in the class next time. We'll probably start by having some open period of time to as an open question and answer in case there are any for the test. I'll put that here. I flip back here. I guess we're going to have, because we don't have class Monday, so the last class before the test is going to be the next class. Okay, so I'll do that. I'll start that class with an open time in case there are things people want to talk about that could relate to the exam. Okay, now we're going to watch a video. I'm going to put the video on slightly faster speed so we can get through it quicker. Now this video is on taste.
We have 5 main tastes. Each taste depends on a particular receptor that's localized somewhere on the tongue. So the 5 different things that we're able to taste are bitter compounds, salty compounds, sweet compounds, sour compounds, and one more thing known as umami. And this is basically the ability to taste a particular molecule known as glutamate. So glutamate. Now these 5 tastes all depend on a particular receptor. So let's imagine that here we have a tongue, and this is the front of the tongue, this is the back of the tongue. We basically have taste buds sprinkled throughout the tongue. So these taste buds are kind of all over the place, and for the most part, they are very, very highly localized in the more anterior aspect of the tongue. So they're more localized in this anterior aspect of the tongue, more so than in the back. There are 3 different types of taste buds. There are taste buds that kind of look like this, and these are known as fungiform taste buds. I'll write that down. Fungiform taste buds. And these are the ones that are mostly found over here in the anterior part of the tongue. There are also taste buds that look like this. where the little buds are over here. And these are known as full-yate taste buds. And these taste buds are mostly found over here on the side of the tongue. And finally, there are taste buds known as circumvallate taste buds, which look a little like this. And the taste buds themselves are actually over here on the sides. And these circumvallate taste buds are mostly found back here in the back of the tongue. Now, each one of these taste buds, if we actually zoom in, so if we kind of zoom in to an individual taste bud, what we would find a little pore. And this pore would have a bunch of different types of cells. So it would have like one cell over here, it would have another cell right next to it, it would have a third cell over here, and so on. And basically each one of these cells is responsible for one of these 5 tastes. So for example, we can take this pink cell over here to be able to detect salty things. And for example, we can imagine that this whitish cell would be able to detect sweet things, and so on and so forth. So the general idea is that each taste bud, so this thing would be a taste bud, each taste bud contains all of the different taste cells. So every single taste bud is able to detect bitter, salty, sweet, sour, and umami compounds. And these taste buds are found all around the tongue, mostly in the anterior part of the tongue. So another way to say this is that we're able to taste all 5 of these different tastes everywhere in the tongue. So it's not as if bitter compounds can only be tasted in this region and salty compounds can only be tasted in this region and sweet compounds only in this region. That's not the case. This is not the case. So it's not like that. Instead, we basically are able to taste all 5 of these different compounds throughout the entire tongue. Most of the taste buds are concentrated over here in the anterior part of the tongue. So what I want to talk about next is something known as a labeled lines model. So here we have a taste bud. As I mentioned before, we have all of the different types of cells, and each one of these cells is specialized to a particular one of the 5 tastes. And what you can see down here is that each cell has a little axon projecting from it. And what is interesting is that these axons actually remain separate all the way to the brain. So all these get projected and they eventually reach the brain. And they actually synapse on different parts of the brain. So let's imagine that this is the gustatory cortex. So let's imagine that this is the part of the brain that receives input from various taste cells. Well, each one of these different axons will synapse on different parts of the brain. So here we've got this axon, it's going to synapse over here, and every other axon, every other taste cell that is this color blue, so let's just say this is a sweet cell. Let's say this is a sweet taste cell. Every single sweet taste cell will send its axon to the brain, and it will all end up in this one region of the gustatory cortex. So we can say this is the sweet region of the gustatory cortex. I keep using this word gustatory, and taste can also be thought of as our sense of gustation, so gustation. Similarly, all the cells that are green, and we can say that green could be bitter, so we can say that green cells are bitter cells, bitter taste cells, they're really bitter. They will all synapse in one part of the cortex, which we can imagine to be the bitter part of the cortex. It's very mad at the world. So this is basically how taste cells send their projections to the brain. They actually send them through dedicated axons, and there's no mixing. And this is known as the labeled lines model. So let me just write that down here, labeled lines model. Again, what the labeled lines model is saying is that each one of these cells has its own dedicated labeled line. And this distinction is basically carried on all the way to the cortex itself. Now let's look at an individual taste cell itself. So let's look at an individual taste cell. So here's the axon. This is the cell nucleus and little hairs that kind of project out into the tongue. So what happens is, let's say that this is a sweet cell, a sweet taste cell. So it's sensitive to sweet molecules. So let's imagine that we've got a little molecule of glucose. So let's imagine you're eating cake, and some glucose will hit the tongue, and some of the glucose will find its way over to a sweet cell. And why is the cell sweet? Well, it's sweet because it actually has receptors in the membrane. So it has little receptors in the membrane that are sensitive to glucose and other sweet molecules. So when the glucose binds to the cell, it triggers a cascade of events that eventually allows the cell to depolarize and send an action potential all the way down its axon into the brain. Now I mentioned that we have 5 different tastes. So we have sweet, umami, bitter, sour, and salty. These top 3 taste cells over here have similar receptors. And these receptors are known as G protein. So G protein coupled receptors. So basically what a G protein coupled receptor is, let me just go ahead and draw it out. It's an intermembrane protein. So this is the protein, and then let's imagine this is the cell membrane. So this protein is a receptor, and the receptor binds to a ligand. So let's imagine that we have glucose in this case. So let's imagine that a molecule of glucose comes in, hits the receptor. The receptor will undergo a conformational change that basically causes a G protein, which is coupled to it. So this is a G protein over here. So when the ligand binds to the receptor, it causes the G protein to dissociate. So this bond is broken and the G protein goes off. And the G protein can do a couple different things inside the cell. So this is inside the cell over here. This is outside the cell. So basically the G protein can go off and do a few different things. Now, one of the things that the G protein can do when it gets dissociated from this receptor is it can actually open some ion channels. So the G protein can go and open ion channels. So the opening of ion channels via conformational change can actually cause the cell to depolarize and fire an action potential. So it fires an action potential. So on the other hand, these two taste cells, sour and salty, rely on ion channels. So instead of relying on a G protein coupled receptor, they actually rely on a receptor. So let's imagine this receptor over here. So in the case of salty, let's imagine that a molecule of NaCl. So a little salt molecule comes in, it'll bind to this ion receptor and then cause the receptor to open up. So the receptor will open up and it'll allow positive ions outside the cell to flow in. When positive ions flow inside the cell, it causes the cell again to depolarize and fire an action potential, and that action potential goes to the brain. So let's look at what would happen if we put a salty receptor inside a sweet cell. So here we have a sweet cell, and the reason that this cell is sweet is because it will have receptors in its membrane that bind to glucose. So let's imagine that we took a salty receptor. So as I said previously, the salty receptors are ion channels, and let's imagine that we put a little salty receptor here. Now, if you remember, the labeled lines model basically says that a particular cell, so in this case, a sweet cell, has an axon that will eventually reach the brain, and when it reaches the brain, since the axon is coming from the sweet cell, will detect the compound that activated the sweet cell as sweet. So what would happen if we put a salty receptor inside a sweet cell? Well, if we have NaCl, so some salt comes in, and it will activate this receptor. And when this receptor is activated, it opens up and positive ions outside flow into the cell. This sweet cell depolarizes and causes an action potential to fire. And by the time the action potential reaches the brain isn't able to differentiate between a sweet molecule or a salty molecule and a sweet molecule. So both salts and sugar will activate the sugar cell. But since the brain has already decided that, hey, this is a sugar cell, and every time this cell normally activates, it's usually something sweet, it's going to think that the salty compound is actually sweet. It's going to detect it as being sweet. So if you were to put a salty receptor in a sugar cell, you can actually trick your brain into thinking that something that is salty, such as NaCl, sodium chloride, was actually sweet.
Okay. I think we'll go to do one more video this time, but not right now. Okay, so a couple of things there that you may have realized. One is that the receptors don't all convert the signal to electrical signals the same way. So there's a category of 3 that converted it based on releasing a protein, and there was 2 others that converted it by opening up and allowing ions to flow through. But in the end, they all have action potentials or electrical signals that are routed to the brain. The model that's used is one where there's a direct connection between each of the receptors and some input to the brain. So we sense molecules that are transported to the taste bud receptors on the surface of our tongue. There's also some on the roof of our mouth and the back of our throat. They're transported through the solid and or liquids that we put into our mouths that we mix with our saliva as we're chewing. So that's how that interaction occurs. The tongue's taste buds has thousands of taste buds in what I'll call a disputed number of distinct types of receptors. In the United States, for many years, it was thought that there was only 4 receptors. This savory umami one is a relatively new one. The interaction of receptors with target molecules results in electrical signals. I put a caution here at the top. How taste sensing really works is still being determined. The receptors are classified based on what is perceived versus what is measured. And so this gets back to what we were talking about before. Unlike the physics, the nice physics of vibrating air or electromagnetic signals, in this case, there's a lot of variables in terms of what comes in contact with the tongue. And what you'll see in most of the research related to tasting and smelling is that they rely very heavily on what the human perception is, or what an animal's perception is. So kind of like the scenario in emotions, where emotion research relies very heavily on what people self-assess their emotions to be in response to things. In fact, you'll see sometimes words related to taste are mapped to words related to emotions, which are in turn mapped, as we saw last time, to words related to colors. So sweet could be happy, sour could be sadness, salty could be disgust, and if you're bitter about something, it means you're angry. So it's interesting that these words have evolved over time to mean things in different spectrums, so that taste can relate to feelings, and colors can also relate to feelings, as we saw last time. We have primary taste sensitivities in molar. So this is the concentration of that substance in whatever liquid it's in. So if you have 0.01 molar sodium chloride, that means you will have 1 in 100th molar. Mole is 6.02 times 10 to the 23rd. And so that means that's how much would be in 100 liters if you have 0.01 molar. You'd have, so those are just concentrations. The receptors for taste behave in what I'll call magical ways, or at least ways that are very different than what we hear and see. This is an example of a molecule that has exact same chemical composition. There's exactly the same amount of nitrogen atoms, hydrogen atoms, protons, electrons. If you weighed these molecules, they weigh exactly the same. They look almost exactly the same, except this portion here in the middle in red. And there, if you see, it's a flipping, basically. The CH2 is attached to the carbon, okay, but this ring is attached to the carbon on the other side. So it's like you just twisted this out of the page by 180 degrees, you have the same molecule. But these two molecules, which look almost exactly the same, except that part is slightly differently oriented, if you put them in your mouth, one of them is aspartame, which is a synthetic sugar, which would be sweet, and the other one has a bitter taste. So to me, this is, if you want to think about how taste works and how different it is, think about the fact that you think about these molecules here, how similar they are, but how different they taste. In a taste test, you're asked basically to taste different types of substances, and in a traditional taste test, they don't have savory or umami because that's a more recent addition. And there you ask for sweet, sour, bitter, and salty to describe those tastes. And then you may also be asked to see how the strength of your taste changes with the amount of the substance you're exposed to. This is a taste receptor. It is actually a device that I invented, but I didn't invent all these applications for it. So on the left, what you can see is there's a cell where fluids can be inputted. So you could take a fan of soda or a glass of water with sugar or something and put it, suck that liquid over the surface. Just like on a tongue, there's biological molecules here that interact with the compounds in that liquid, and depending on their affinity and their binding, they change what's called a surface plasmon here in this device that then can be read out electronically. So you can make artificial taste sensors. Matching them to what a human tastes, of course, is more of a challenge. Okay. Now we're going to get into smelling. I'm going to try to show this video really quick. We might cut it a little short so I can show the rest of the slides. We did start a little bit late. If you guys can hang on a few minutes later, we'll get to a nice clean endpoint.
You've probably noticed that whenever you have a cold, your nose gets all stuffy. And when your nose gets all stuffy, with a bunch of stuff, you're not able to taste things very well. So let's imagine that you're trying to eat a strawberry. So you're eating the strawberry, normally it tastes really good, and it goes in your mouth. And as you're chewing the strawberry, you're breaking down different cellular components, and these cellular components release little molecules. And these little molecules will travel through the back of your throat, and some of them will actually come into your nose. And as you're chewing the strawberry, you're actually smelling little molecules that are being released. You're actually using your sense of smell in conjunction with your sense of taste, with your ability to taste the strawberry. And so if you knock out your sense of smell, which is what happens when you have a cold, if your sense of smell is knocked out, you're relying only on your sense of taste. And when you only rely on your sense of taste, you're not able to taste things as well. Things don't taste as rich. So you can actually try this. Next time you're eating something, just go ahead and close your nose and see if whatever it is you're eating tastes differently after you're no longer smelling it. So hopefully this goes to show a little bit about why our sense of smell is important. Now, we can definitely live without our sense of smell, but it does greatly enrich our everyday lives. So smell is also known as olfaction. So sometimes our sense of smell can be called our olfactory sense, and we'll talk a little bit about how this word comes into play and a little bit of the anatomy later on. So let's go ahead and go into the anatomy of our olfactory system really quickly. So if I, again, draw a profile of someone, here's the nose, here's the mouth, and here's the chin. There's a nostril, and this nostril basically is an opening to allow air and various odor molecules to come into the nose. And there's actually a region that you can't see, but this region is known as your olfactory epithelium. So this region is the olfactory epithelium. Olfactory epithelium. So again, olfactory, olfaction. So separating this olfactory epithelium from the brain, because the brain actually sits up here. So we've got a little bit of the brain and it sits up here. So separating the brain from the olfactory epithelium in this nasal passage over here is a piece of bone known as the cribriform plate. So this little piece of bone is known as the cribriform plate. And sitting right above the cribriform plate is actually a little extension from the brain. So there's actually an extension that comes from the brain, and it sits right above the cribriform plate. I'm drawing it in here in purple, and it looks like a little bulb, and this is actually known as the olfactory bulb. So olfactory bulb. So again, olfaction, olfactory bulb. And the olfactory bulb is basically a bundle of nerves. So it's actually one of the cranial nerves that exits the brain. And so this bundle of nerves sends little projections through the cribriform plate into the olfactory epithelium. And so this projection of nerves breaks off and branches off, and there are thousands and thousands of cells sending little connections into the olfactory epithelium. So I'm only drawing a few of them over here, but there are thousands of these cells sending little connections into the olfactory epithelium. And at the very end of each one of these little cells are receptors. And each receptor is sensitive to one particular molecule. It's actually sensitive to one particular type of molecule. So for example, let's imagine that we have a benzene ring, and most things that have benzene rings are known as aromatic compounds, and normally you can actually smell aromatic compounds. So if a molecule has benzene ring, it usually has some sort of scent, and that scent is picked up via the olfactory bulb and these little extensions that come into the olfactory epithelium. So we have a little benzene ring, a little odor molecule, and it travels into the nose, and it'll actually bind to a small receptor on one of these nerve endings. So what I'm going to do next is zoom in into this region over here. I just want to look at the olfactory bulb, and in particular, I want to be able to look at each one of these little olfactory sensory neurons and their little projections that bind to various molecules in the environment. So what I'm going to do now is zoom into that little part that I highlighted earlier. And so we're just going to look at the olfactory bulb. So let me just label this olfactory bulb. And as I mentioned before, the olfactory bulb was separated from the nasal passage by something known as a cribriform plate. This cribriform plate is basically just bone, and there are a bunch of little holes in the cribriform plate that basically allow the cells, these olfactory sensory cells, to send projections to the olfactory bulb. So let's imagine that there is a olfactory sensory cell, and it sends a little projection into the olfactory bulb. And so it sends a little projection. And this cell is situated inside the olfactory epithelium. And there are all kinds of other epithelial cells over here, so I just kind of draw those in. So there are all kinds of epithelial cells that basically secrete mucus and all kinds of things. And these olfactory epithelial cells are situated in between the rest of the epithelial cells. And there are thousands of different types of epithelial cells, and each type has a particular receptor. So let's say that this particular olfactory sensory cell is responsible, has a little receptor, and this little receptor is sensitive to benzene rings. So as we have a benzene molecule enter the nose through the nostrils, it enters the nasal passage and eventually will bounce into this receptor. And when it binds to this receptor, it will actually trigger a cascade of events that causes this cell to fire. This cell will fire an action potential, and the action potential will actually end up here in the olfactory bulb. And what we have is we actually have a whole bunch of olfactory sensory cells that are sensitive to the same molecule. So we have another one over here, and it will actually synapse to the same place. So we've got another olfactory sensory cell, and this also is sensitive to this benzene molecule. So benzene molecule comes in and activates a whole bunch of cells that have benzene receptors, and all the cells that are sensitive to benzene will all fire an action potential to one particular location in the olfactory bulb. And this particular location is known as a glomerulus. So glomerulus. And the glomerulus is basically, we can think of it as a destination point for various sensory olfactory cells that are sensitive to the same molecule. So we could think of this glomerulus as a benzene glomerulus because all the cells that are sensitive to benzene will synapse over here. And what actually happens is they will synapse onto another cell type. So there is another cell, and this is known as a mitral, so mitral or as a tufted cell. And this mitral or tufted cell will actually project to the brain. So the reason we have this type of organization is because it's a lot easier for one cell to send a projection to the brain than it would be for thousands of these cells to send a projection to the brain. So just to kind of go into this in a little bit more detail. So what we can also have, sorry about that. What we can also have are other cell types. So let's imagine that there's this blue cell over here. And let's imagine that this blue cell is sensitive to two benzene rings that are connected to one another. So if we have two benzene rings that are connected to one another, they will bind onto a receptor on this blue olfactory sensory cell. And this blue cell will go through the cribriform plate and synapse in the olfactory bulb, but it will synapse at a different location, as will all other blue cells sensitive to double benzene rings. And they will synapse onto a double benzene glomerulus, and so on. And so this double benzene glomerulus has a different mitral or tufted cell that will send a projection to the brain. So this is the general organization of various cells in the olfactory epithelium. And the general idea here is that we have hundreds of various cells that are sensitive to one particular molecule. And these hundreds of cells will all send all their projections to one glomerulus. And that one glomerulus will have several mitral or tufted cells that will then go to the brain. So now what I want to go into is I just want to look at this part right here. So I want to look at how a molecule will bind to a receptor and how it triggers an action potential. So let's imagine that we have this molecule of benzene over here. So it will enter the nasal passage and it will bounce around until it binds to a particular receptor. So what we have is a particular receptor that is in the membrane of a olfactory sensory cell. So this benzene molecule will come in and it will bind right here, it will bind, and when it binds, it will actually cause this receptor, which is known as a G protein coupled receptor. And it's called a G protein coupled receptor because there's actually a protein inside that is known as a G protein, which it is coupled to. So it's coupled, it is coupled, the G protein is coupled to the receptor. And when the molecule benzene binds, it will actually cause dissociation here. The G protein will break away and it will cause a cascade of events inside the cell. So this is inside one of the sensory olfactory cells that we were talking about earlier. And so one of the effects that this G protein will have is it will actually bind to an ion channel. So it will bind to an ion channel. And the ion channel will basically allow positive ions from outside the cell to float inside. And this will cause the cell to depolarize and fire an action potential that eventually will go through the cribriform plate and then synapse to a mitral tufted cell. And that mitral tufted cell will send a synapse to the brain. So let's just go over everything we talked about really quickly. So we have a odor molecule. So an odor molecule, which is a benzene ring in this example, will bind to a GPCR. So the GPCR is a particular odor receptor. And the G protein, so the G protein coupled receptor, the G protein will actually break away. So G protein breaks away and activates various things inside the cell. And one of those things is an ion channel gets activated. That causes the cell to depolarize and fire an action potential. And the action potential will go through the cribriform plate and into the glomerulus, and it will activate a mitral or tufted cell, which will then enter the brain. And then your brain is able to perceive that odor through this pathway.
Okay. Both those videos, you're going to want to probably review them again. Links to them are available on Moodle, so you don't have to pull up the whole class presentation. You can just go directly. They're underneath where the McGurk effect is. So I'm just going to go through a few more slides because I think it's not going to take more than 5 more minutes. Just to give you a better idea of what was, or add a little bit more to what was in the video. If you can look here, this is just one of many examples of compounds that we can actually distinguish with our nose. We have a lot more receptors in our nose. And so we can distinguish lots of different types of compounds. This is similar to what I showed you for taste, where in this case there's smells, but the exact same scenario, where the atomic weight's the same, chemical composition's the same. It's just the orientation's a little bit different. So these are flipped, if you will, out of the page 180 degrees to create this. But this you'll smell as spearmint, and this you'll smell as caraway. So two very different and distinct smells. Now here's where it gets exciting. We smell combinations of molecules transported in air, as opposed to taste, where they're physically either in a liquid or a solid, generally. The nose has 5 million receptors. That's why if you call in the middle, whereas in the taste, each one sort of channeled its way to the brain, here each type gets grouped, and then their response is channeled to the brain. So they sort of sum up the response of all of that one type. This is somewhat developing, but there's an estimated 400 different types of receptors. If we want to put that in context, we saw there was 5 different types of taste receptors. And then remember in vision, there was 3 different colors of cones. And in sound, there was 1 vibrating membrane. So, you know, this is a lot of information with 400 different types of receptors that combine, and scientists estimate, to allow a human being to distinguish up to 1 trillion scents. That's a lot of scents. So this is what I call classic pattern recognition and learning. The information coming in is not well organized. There's not, good physics equations that are telling you what's happening. If you put something in your mouth, what you taste will actually change over time. And so, so there's a lot going on here in the brain. Really, the brain has to make guesses. It's not like the sound and the sight where things come in well organized and the brain just has to see what's there or interpret what it's hearing. Here, it has to basically make guesses to make any sense at all. So we associate smells with experience sometimes. We certainly don't have as many words as would be necessary to describe all the scents that we have. In fact, you can see there, if we took 1 second to write down each of the trillion scents, it would take us 31,000 years to do it. There are smell tests. I'll let you read through this later. Basically, scratch and sniff is a smell test, and they have some that tell you how sensitive you are to smell. This is what an artificial nose can be comprised of. I actually worked with a group from Caltech back 20 years ago who was making an artificial nose to detect buried landmines. Here what they have is a chemical sensitive layer that's on top of a substrate that vibrates. The substrate's made to vibrate. And as things bind to that chemical sensitive layer, it changes the mass of what's on that piezoelectric substrate, which causes the vibrational frequency to change. So when something binds, changes the mass, it increases the mass, which lowers the vibrational frequency, which is read out electronically. And the idea here is that you create a whole bunch of chemical sensitive layers that are just sensitive to different things. You may create something that's more sensitive to benzene, or you may create something that's more sensitive to double benzene rings, and so forth. And in both cases, for taste and smell, we can see how that chemical input put into an array can then be interfaced into electronics, conditioned, and then PARC is a pattern recognition. So pattern recognition means it's ripe for AI. So you can see that to the degree that you have sensors that can distinguish lots of different types of basic senses, smells or tastes that you can envision not only making an AI, but you can envision requiring an AI to make sense of what's incoming. And so to some extent, again, because this isn't physics and it's chemistry, and because these receptors have varying degrees of receptivity, and there's so many of them, we require our brains really to use their neural networks to make sense of what we're tasting and smelling. So next time we're going to pick up with the last slide. We're not going to do this now, but this is where we compare and contrast the eyes and ears now to the tongue and the nose. And that'll be a good review for what we did today. Are there any questions before I end today's lecture? Okay, well, if not, remember I'm going to for sure have the homework number 3 up before tomorrow midnight, but I'm definitely going to try to have it there today. If you do have any other questions, that's a good point. Just put them up on Piazza. I went through the videos fairly quickly. You can play them in slow or their regular pace by just finding them at the bottom of the Moodle page where they already are. Thank you once again for joining and helping us keep on track for the semester. Take care. Have a good evening.